All right, here we are with chapter 15. I am going to break this up into two lectures. They both are included on the PowerPoint I've posted for you. The first one will be about cell-cell junctions. The second lecture will be on the extracellular matrix. Okay. So first we're going to talk about different ways cells can interact with one another. And you see a nice list here for our cell-cell junctions. This is really important in eukaryotic cells in order to make coordinated efforts with cells as things like tissues. Okay. So the first type we're going to talk about is adhesive junctions, and there's adherens and there's desmosomes. So adherens are shown here. Their primary transmembrane protein is called a cadherin. And cadherins, the Ca part of it, Ca, tells you that calcium is important in helping these proteins interact. So you have one cell here and you have one cell on the left, and they're connected in the middle by the E cadherins. Then you have all these linker proteins. Linker proteins. And then you have the cytoskeleton. So in this case, adherens connect actin to the plasma membrane. This is your plasma membrane. To another cell. And there are different types of adherens in different types of tissues. One of the terms that you are asked to learn for this chapter is homophilic interactions, right? So homo means same. If you've taken genetics, you've learned homozygous, same alleles. Philic means like, so you've learned hydrophilic, right? Water loving. Okay, so these things like to bind to the same type of protein. And that's really important when you were, you're not going to be anymore, you were a little embryo developing, and you have all these different cell types. And so in order to help organize them into tissues, for instance, all the ones that expressed E cadherin could start interacting with each other, and ones that produced P cadherin would interact with each other. So it was a way for the early embryo to link cells, like cells, into tissues. Okay. Adherens are found as a continuous belt around the apical side. So up here, when we talk about things that are polarized, I really hate it when it does that. Polarized, right? Polarized means it has two different ends. So an apical side, and down here would be the basal side. And this is quite characteristic of epithelial cells. So you have a kind of like a top layer and a bottom layer. Versus you could imagine like a 3D organ that would not necessarily have different ends to it. Right? So adherens, you can see here, are formed as this belt kind of around closer to the apical side of a cell layer. Okay. The next type of adhesive, so adhesive means we're linking cells together, okay, and we're connecting them. This type is called desmosome. Looks very similar to an adherin junction. It has special types of cadherins called desmoglians, desmocolins, right? But the same idea, you have this transmembrane cadherin protein, you have linker proteins, and then desmosomes are attached to intermediate filaments. That's different than adherins attached to our microtubules or actin. These are found as what I like to call spot welds. Right? So they're these button-like, really strong adhesive, helping the cells stick together, function as a unit, resist stress. And this is showing a desmosome that we have something called a plaque. And so a plaque means a whole bunch of the desmosome proteins, right, the cadherins, the linker proteins, interacting with tonofilaments. That generally we know them as intermediate filaments. Right? And so versus adherins, where you have a few here, desmosomes have this thick, like I said, like a button or a spot weld. So it's this big, thick junction that holds these two cells into place. So both adherins and desmosomes are types of, oh my gosh, I just lost the word, attachment junctions, right? Anchoring, there we go. Anchoring junctions, so desmosomes and adherins. All right, so these are holding our tissues together, our cells together into tissues. Then we have something called a tight junction, and that's right up here. And tight, no, not tight junctions. I'm sorry, not reading my slide. Transient cell junctions. These are tight junctions up here, but that's not what I'm supposed to be talking to you about in this slide. What I'm supposed to be talking to you about, sorry, are these transient. So these are much less strong junctions. They help promote cell-cell adhesion, but they're more generalized. Right, so selectins, the other one that's not in here is lectin, and here's the CAMs. So they're binding, but they're not for adhesion, and they're not, as we'll talk about, gap junctions for communication, and they're not for a barrier. So they're more, we call transient, they can come and go. Selectins is an example of one of these transient cell-cell interactions. And I don't think I said here, but this is a lot of carbohydrates, sugars. And you probably know that if you get sugars wet, they get really sticky, right? Think of syrup. Think of honey, right? So these things are helping cells stick together, and they're transient, right? You can put honey between your fingers, go do this tomorrow and eat breakfast, right? And you can break your fingers apart, and then you've got this gooey mess. You know, science is fun. So selectins are glycoproteins, and glycoproteins just mean proteins with sugar on them, and they can interact with other carbohydrates or sugars on a cell. And that's trying to show you right here. So you've got this glycan selectin. Here's a glycoprotein from the endothelial cells, and this leukocyte, is a white blood cell. You don't want all your white blood cells swept away in the blood, right? Here's your blood flow. You want them to be able to stick to get to where they want to go. And there's a term here that you know now called chemotaxis, right? So this site of injury is sending out little chemical messages that say, help, help, come help, I've got an injury, or I've got some kind of invading bacteria or virus or something like that. And so this white blood cell is producing these sugars, and we've got selectins on the endothelial cell, and so the white blood cell rolls and interacts and rolls and interacts, and it's an interaction that's not permanent but strong enough to keep it being swept away in the blood. And then when it finally interacts with like an ICAM or an integrin, it becomes more stable, and so now the white blood cell stops moving, and now it can go squeeze between that endothelial cell layer and go help at the site of injury. Tight junctions. This is where I thought we were before. Okay, tight junctions form barriers. Okay. It prevents movement across a cell layer. Okay. They are so tight that they can prevent fluids, right? So it's this really super tight junction right here. I'll move that in a minute. And it blocks even liquid movement. It's amazing. Tight junctions are made of proteins, transmembrane proteins called claudins. So you can kind of see the word claudin here, and there's O-claudin, right? And they're interacting with actin. And you can see that claudins have this characteristic looping structure. What's really cool is, even though they're tight, they can allow some ions and small molecules through, but they're going to prevent things like big proteins, right? So this is really important, say in your bladder, in your pancreas, in the lining of your gut, right? You don't want digestive enzymes to be able to move through. So it's stopping movement this way, and it's stopping movement that way. Okay, so both types of movement. I'll get all this stuff off. Okay. What you don't want is this barrier to be broken. Okay, so you don't want to lose the integrity of your tight junctions, because things as big as bacteria and proteins and viruses, like it says dietary antigens, those things can then cross that epithelial cell layer and cause issues in the tissue down here. Right? You don't want the digestive enzymes from your gut to start digesting molecules outside of your gut. So that's an ulcer. Okay. An ulcer is a break in this epithelial layer where enzymes, bacteria can get to where they shouldn't be. Another thing illustrated on this slide is paracellular transport. So paracellular means between the cells. So right here, or right here for small ions, small molecules, versus going through a cell, right? We've talked about endocytosis to exocytosis, things like that. Paracellular is that movement between cells. Okay? So you want to be able to control this, and you don't want that to happen. Okay. Another type of cell-cell junction are called gap junctions. And gap junctions are like little tunnels. Now, gap junctions are really small, like only about 3 nanometers in diameter in this open space for things to get through. So ions, small molecules, single sugars, single nucleotides, those kind of things can go through. And this is allowing these cells to communicate. Okay. This is really important in the heart. So you know that calcium is important for muscle contraction. And so the way the heart cells beat, or the way the heart organ, I should say, beats, is the heart cells pass calcium through gap junctions from cell to cell to cell. And so you get this movement of the heartbeat. If you want to see something cool, click on the video here. It's talking a little bit about stem cells and how we can make heart cells from stem cells. It's like a 30-second video. But it shows that telling a cell in a petri dish that is a heart cell, they will make these gap junction connections and they will send calcium from cell to cell. And in the petri dish, you can get this beating, I can't draw beating little clump of cells. It is really, really cool. So this is for communication, rapid communication. These cells are in direct chemical and electrical contact. The similar type of junction in plant cells, and the only type of stuff outside a plant we're going to talk about, is the plasmodesmata. So these are like big gap junctions. And when I say big, they're big, like 20 to 200 nanometers. So they're really big tunnels. So they connect cells, plant cells, and this is important because remember, plant cells have cell walls, right? So they have this extra rigid outside the plasma membrane. So they really need these tunnels, these plasmodesmata, to be able to talk to one another. And they're so big that you can get membranes across. And desmotubule is an extension of the ER from plant cell to plant cell. Okay, so I hope you can see this membrane, and I'll show you another picture that shows it a little bit better. Well, let's just go there. So I turned this picture on the side so we're keeping the same orientation. So what I want you to see is that this big space is the plasmodesmata. Here's this big tube or a tunnel. Okay, and let me get rid of all that. Okay. Here's the plasma membrane. So the plasma membrane from one cell to another is continuous. Okay. Outside here, cell wall. Okay. So plants share a cell membrane. This purple is the ER. And you can see that the ER membrane can go between from plant cell 1 to plant cell 2 through that plasmodesmata. So this part is called the tubule. Let's see if I can change. Hold on. I wish there was an easier way to do this. Okay. This space that I'm coloring yellow, okay, that's called the annulus. And you can't read that, so I will, where'd I go? Pointer back to my pen. Annulus. Oh, it's getting glitchy. Oh, dang it. weird. Okay, so that's the space. And then what I've drawn here in this picture are viruses. So plants can get viruses just like you and I. Viruses have to get through a plasma membrane. So for us, a lot of viruses come in via receptor-mediated endocytosis in clathrin-coated pits. Plant cells don't do that because they have that cell wall, right, on the outside that's preventing things from getting through. So the way a plant virus gets through is usually by an aphid or a bug feeding. So this is my aphid. Right, I don't know how to draw an aphid, but it feeds and it's sucking cytoplasm. And when it does that, it can deliver these viruses into the cell. And now the virus can travel cell to cell by going through the plasmodesmata. And cell viruses can be between 20 and 50 nanometers. So they definitely could not fit through a gap junction. So we do not have viruses transmitting from cell to cell through our gap junctions. But plasmodesmata are definitely big enough. And some plant viruses can actually stretch this even bigger if they need to go through cell to cell. So remember that plasmodesmata is my favorite biology word. You will definitely see it on quizzes, exams, and final exam. You've been warned. Don't forget it. Okay, last slide for this lecture, and then I will take a break. Homophilic, heterophilic interactions. Right? So again, we talked about homo means same, hetero means different, like heterozygous in genetics. Okay? So some proteins, like cadherins, have homophilic interactions. They only like to interact with something that is similar. And this is showing you some ICAMs. Some proteins, or proteins we'll say, have heterophilic. Okay, so for instance, integrins, which we're going to talk about in the next lecture, and fibronectins. When you have things that are different, right, they're hetero, different. So heterophilic interaction just means between different proteins or different molecules. Okay, so here's a CAM and a selectin. All right, so different types of molecules prefer heterophilic, homophilic, and some do both. So just really make sure you understand these terms, and you can come back when you're ready and we will talk extracellular matrix.